I would like to invite Praveen Prathapan with his lecture on social protein thinking.
Thank you very much. Hello everyone, my name is Praveen Prathapan and I'm a medical writer from London. This is my first Biosemiotics gathering and you've been so kind and lovely, so thank you so much for having me. I have two apologies to make. One is I'm not going to talk about time like I was going to in the abstract. Instead I'm going to talk about the genetic code itself. And the second apology I have is I'm going to talk about an enzyme called aminoacyl-tRNA synthetase. And aminoacyl-tRNA synthetase is quite a mouthful to say. So I'm going to abbreviate it to the official abbreviation, which is AARS, which is pronounced ARS. For the next 15 minutes, your first impression of me is going to be me talking about ARS for the next 15 minutes. Alright, so yeah, my title is Social Proteins and I'm going to talk about the genetic code. And to start with, I'm going to invoke Evelyn Fox-Keller, who said, "It seems evident that the primacy of the gene as the core explanatory concept of biological structure and function is more a feature of the 20th century than it will be of the 21st. And of course, this is an allusion to the selfish gene concept of Richard Dawkins, which I grew up with in my biochemistry training, and is perhaps the paradigmatic example of neo-Darwinism that I suppose we are all opposing. So how do we oppose that? So today I'm going to talk about what the genetic code is. I'm going to introduce ARSes, and then I'm going to introduce the ARS ribosome loop, and then finally I'm going to talk about implications for normativity. So what is the genetic code? The genetic code is the set of rules used by living cells to translate information encoded within genetic material into proteins. But already I have a problem with this. I find this misleading because it's not a set of rules. Ontologically, the genetic code is not a thing. And I'm going to use Yuri Lotman's quote from 1990, where he said, "If we have to use a metaphor in order to imagine the capacity for memory, then the least appropriate one is the image of a library with books in its shelves, or a computer with data of whatever quantity stored in its memory. Memory is more like a generator." And so the genetic code is not a thing, a process view of the universe. And I met a couple of Whiteheadians yesterday at the pub, so I'm with you here. A process view of the universe, and particularly biology, requires a reframing of certain entities as fundamentally processual. And John Dupre and Stefan Göttinger use viruses as living processes. In fact, I would go a step further and say viruses are semiosic processes. So going back to the genetic code therefore is not the set of rules, but the setting of the rules, a generator. It's ontologically processual. And that brings me on to aminoacyl-tRNA synthetases. These are essential enzymes found in every single cell on Earth. And what they do is they catalyze tRNA charging. And so what they do is they literally ligate enzymatically tRNA molecules to the cognate amino acid. And that's a biochemical term, cognate amino acid, the correct amino acid. Correct for whom? I'll talk about that later. And so last year I wrote a paper in the European Journal for Medical Genetics, where I denoted tRNA as representamen, the cognate object as the amino acid, and the interpretant being ARSes, these proteins. And there's a quote from Terence Deacon, but I'm sure loads of people have said this: interpretation is a process that requires work. And so this process, this enzymatic catalysis, requires ATP. The energy currency of the cell is needed to actually enact this reaction. So I wrote this paper and I felt pretty proud of myself. And but then I started having a few sleepless nights and I could hear Kalevi's voice in my ear. No semiosis beyond Umwelt. I had not invoked Umwelt at all in this paper. So I was really kind of struggling. This model is incomplete. It misses Uexküll's functional circle. Otherwise, you could argue it's just a chemical reaction. So what do? It turns out ARSes are what is known as the chicken and egg protein. Loads of people have talked about the fact that this protein, which enacts the genetic code, is itself made by the genetic code for which it enacts. And so it's in a loop with the ribosome, as I've denoted here. It's a bidirectional loop. And this is what people like Carl Woese have called a chicken and egg paradox. The evolutionary history of the current aminoacyl-tRNA synthetases, says Carl Woese, must go deep into translations past to the emergence of the modern genetic code. Peter Wills has talked about this more recently. He said the essentially autocatalytic character of their production is an outcome of the interpreting reflexive sequence information in part of the cell's genome template. And he said this in the Biosemiotics journal in 2014. And that's an interesting term, autocatalytic. I think this arse ribosome loop is a vindication of Howard Pate's semantic closure or epistemic cut. I would also say that Rosen's closure to efficient causation is also vindicated by this idea. The catalyst needed for its operation must be generated internally, and to map it onto the MR system, ARSs can be said to metabolize the genetic code, and ribosomes repair back the ARSs into the system. And I would also say this is reminiscent of Kaufman's autocatalytic set, which is in line with that Peter Wills idea that I said earlier. And based on Jaime's work, I would even say this is semio-autopoietic. I wouldn't say this is info-autopoietic because it's a process. The ARSs are always enacting the genetic code into being, bringing these two genetic moieties together. So to make this a model, I can't help but see that on one hand, we have the ribosome, which is, I think it's a misnomer to call it a translator. I think the translators are the ARSs. They're the ones that are actually putting the cognate amino acid to the tRNAs, whereas the ribosome is just printing out proteins. And so that's the vehicle, that's action, it's printing out proteins. And I believe that ARSs are doing the perceiving side, the semiosic side. And so I think this is reminiscent of some sort of Funktionskreis, some sort of genetic Umwelt perhaps. But one thing that's not really talked about is that the ARSs exist in a consortium. There are almost always 20 ARSs in every cell on Earth. The 20 ARSs actually comprise two evolutionarily unrelated classes, class 1 and class 2, which implies a symbiogenic origin for the genetic code. And of course, Lynn Margulis, I think, will be forever vindicated. We're going to be discovering more things that are symbiogenic, I think. ARSs are also, in biochemical terms, a quasi-species consortium, or a large group or cloud of related genotypes. And I'm going off the definition by Louis P. Villarreal from 2021. And even Peter Wills and Charles Carter have used the term quasi-species to understand aminoacyl-tRNA synthetase enzymes. And so I would call this a minimal society, a set of agents whose actions are constrained by shared rules, such that violating those rules destroys the group that sustains them. That's why I think there are implications for biological normativity. Normativity is rules. It comes from the Latin word norma, which actually meant the carpenter's T-square. So a right, which denoted the right angle, not the wrong angle, the right angle. And so normativity concerns the standards of what ought to be done. So the most famous person who talked about that, I suppose, is Hume. He talked about the is-ought problem, which states that it's not obvious how one can transition from descriptive statements to prescriptive statements. So he would have thought that normativity biologically arises in humans. So he would be utterly shocked by this presentation. Of course, normativity is in Nook School. This is just here for illustrative purposes that he talked about rules all the time. Of course, more recently, normativity has been talked about by Kuhl, Deacon, Emmeke, Hofmeier, and Steinfeld in this amazing paper, where they said that normativity is associated with preserving the system. I'm going to invoke Thomas Sebeok now, whose axiom was that semiosis is the criterial attribute of life. And I would say that this presupposes normativity as a criterial attribute of life. And I'm not the only one. Our next host for the Biosemiotics gatherings, Tom Frazee, said that to understand the origin of life, we must first understand the role of normativity. And so I would say normativity is associated with the origin of life. I'm not alone here. The other thing about normativity that I hinted at before is that it's shared rules, and so it's social. For example, disciplines like sociology and anthropology analyze social norms as shared and enforced rules that regulate communal behavior. And so if we go back to our model of the genetic code, I would say, is a consortial processual normative loop. And here I've changed the R's to the 20 R's. However, even this is not enough. We need to open the loop. And in this new book that's coming out, Charles Wolfe wrote the third chapter, I believe, and in it he said, the Bowel plan concept in Uexküll's theoretical biology is ambiguous. The idea of a conformity to a plan seems to fix the organism to its environment. It is unclear whether this ambiguity was ever fully resolved in Uexküll's work. Nevertheless, Goldstein and Canguilhem nudged this in a less fixed direction. And so now I'm going to talk about the main person of my presentation, which is Georges Canguilhem. He was a physician, actually he was a philosopher, he studied philosophy, and then in the 40s he became a physician, and he worked in the French resistance against Germany in 1943. And that's when he published his seminal work, Le Normal et le Pathologique, in 1943. And then after the war, he came back to being a philosopher and ended up becoming a professor of philosophy in France in, I think, the 50s. I mean, this is 20th century France, professor of philosophy. He is it. In fact, he was a doctoral advisor to Foucault and Simondon, so I see a little lineage here. Foucault, who wrote the foreword to this book, said that Canguilhem's was a philosophy of error. He viewed living systems as actively self-produced, and in fact, I've seen two sources, I couldn't believe this. He coined autopoietique in 1951, according to these sources. I need to read the original, but that blew my mind. And I would even go a step further and say he's anti-Barberian, because he recognizes molecular misinterpretation. And I would also say that he's a crypto-biosemiotic, but he came at it through normativity. And so here's the quote I pulled. If, in principle, organization is a kind of language, the genetically determined disease is no longer a mischievous curse, but a misunderstanding. There are bad readings of a hemoglobin, just as there are bad readings of a manuscript. But here, we are dealing with a word which comes from no mouth, with a writing which comes from no hand. So how does he open up this loop? He introduces the idea of vital normativity. Canguilhem was actually a self-proclaimed vitalist, which is scary. I'm scared to say this. But yeah, he said that organisms create norms. The healthy state, says Canguilhem, allows transition to new norms. Therefore, disease would be considered the narrowed or diminished margin or capacity for normative activity. And Thomas Osborne, writing in Biosemiotics in 2016, said that contemporary vitalism should see life as not merely process, but as norm-generating vulnerability. And so how can this feature into our model of the genetic code? Well, the thing about the genetic code is it generates other proteins. It doesn't just maintain itself, it creates non-RS proteins. In fact, it also creates proteins that reinforce the ribosome structure itself. And so it's not only self-maintaining, it's creating new codes. So Barbieri has a happy ending because new codes are generated in this model. And so it's fundamentally creative. So the genetic code is a norm-generating, consortial, processual, normative loop. So to conclude, the genetic code is a process, Rs-catalyzed triadic semiosis. The genetic code requires work, ATP-dependent semiosis. The genetic code is consortial, it requires 20 Rs. The genetic code is looped, the Rs ribosome loop, which engenders normativity, error, mistakes, a point of view, Umwelt, etc. And one thing I didn't say is that the genetic code is context dependent. For example, amino acid availability is dependent on that, and therefore it's indeterminate. And finally, I would say that the genetic code is norm generating. It generates non-RS proteins. And so, if we go back to Evelyn Fox Keller, if 20th century biology was characterized by the selfish gene, perhaps 21st century biology can be characterized by social proteins. Thank you.
Okay, thanks, Praveen. Very nice. Don't you think this is closer to code biology than biosemiotics?
A code sounds like a thing. That's the first problem I had with it. A code does it have misinterpretation? I don't know.
Well, you're acquainted with Poirier's work, right? To him, the tRNA functions as an adapter. It functions as a kind of what he calls a join between two different worlds, right? Code maker, the nucleotide sequence and its so-called meaning in the amino acids. To put one brief way of getting at it is, do you think this code is generally semiotic and that it has the capacity for the equivalent of choice and creativity, or is it more like a fixed program that may have been semiotic at one point, but is now more or less, for all intents and purposes, deterministic?
I think it's constrained, more so than our language, for example, the codes of culture, as Barbieri would say. But I think it's also amenable to change. Mitochondria, for example, have different codes. There are some bacteria that have different codes. Why is that? I think it's because it was amenable to change, and perhaps still is.
Excellent paper, of course. Just clarification about context dependency of code, as you said. You can put back that last slide, please. That's informative also. You understand that in the case of any chemical reaction, where the reactant in principle can have reaction with more than one substrate. Unorganic reactions can also be like that, you understand? So the product will be different accordingly. We certainly would not call that context dependence. So I wonder whether here, in case of genetic build processes, it's just the same case. That this is a competition between two sorts or more substrates, and this is not real context dependence, which presumes a relation of, relationship.
Yes. I actually say that it's not so much the problem here is declaring the genetic code as indeterminate isn't correct, right? You could have two different
So this is a relation. This is other aspect of the same thing.
Thank you.
Two. Your responses?
My response is, I think that is the most contentious part, and that wasn't in my presentation. I added it on at the conclusion, and definitely something for me to think about.
Okay.
One way to think about that is, since you're already invoking Simondon, at least by lineage, if you could discern and elevate the asymmetry between the two sides and talk about how, I really like that. I think your intuition is right. This society, this idea that you have the 20 of these things, but distinguish further how it is they're distinct. So, for example, like from the the more stable operations of the RNA. So, for example, Simondon might suggest that society, so to speak, it has access to the pre-individual charge. It's fundamentally metastable, and there's different physiochemical ways you could explore that. For example, the Kuramoto model. I know you've read a paper or two of mine, I think, but if you look at, I invoke the Kuramoto model as a large population of diversity tuned chemical oscillators, which basically are catalytic sets. And those are in a milieu, a continuous milieu, and you have plenty of opportunity there for indeterminacy and generativity and true contextuality, because that milieu can be sensitive to our arbitrarily many perturbations. And then as Simon talks about is that harvesting this, well, I call it harvesting the sparse order from the noise. Simon will talk about as a disparation, that you have all these degrees of freedom within those 20 interacting arses, and that all those degrees of freedom interfere, generate noise, and that is a source that's fundamentally distinct from what's happening on the other side of that cycle. So I think you're on the right track, but yeah, keep further.
Thank you. I would say just, yeah, I don't know much about Simon though, but I need to learn. One thing about them being distinct, one thing about the two classes of arses, class 1 and class 2, is that one actually is to do with hydrophobic amino acids, I believe, and one is to do with hydrophilic, so that might introduce some sort of thermodynamic.
Well, and tension.
Yeah.
Thank you.
Oh, yes.
I liked your presentation very much.
Thank you.
Related to what Kari said, I have the impression that genetic code you are talking about is somewhat more flexible than used in the literature, like biology, etc. So you're somewhat containing in it, including in it, the more flexible aspect, metastable component, as far as I can recognize what you are writing. So perhaps with that tuning, alignment with the terminology in the literature would be less controversial. I like it very much, and I also see parallelisms to my presentation in the sense that the codes are the sediments to refer to some notion merely has coined previously, I think last year, of the individuation process that conserve both qualities or structures and relations, which of course not necessarily exhaust the whole pre-individual charge. In that sense, they are conserved and serve as the backbone, as the scaffold for further individuation. Thank you.
Thank you.
Thanks so much for your talk. Two things. One is, would habit be an auspicious substitution for norm, which is sort of heavily laden? Yeah, that's one thing. The other thing is, we are now speaking in the English language, and it is speaking to us. And each language, everyone knows without reading George Anderson and his interpretation, has its own personality, positive and negative constraints. So we are enabled and limited by a hero-oriented language that is heavily, compulsively using and creating and borrowing and stealing nouns, which are things. So maybe it's not our fault that we have been thinking of things. It is shaped a little bit, not determined, but shaped a little bit by so much of our discourse in this arena being in English.
Yes, thank you. Yeah, when I tend to think about process philosophy, it's facilitative constraints that cause the eddies in the whirlpools, for example. That's how I see that there are, of course, things, as we say. But I like the idea of positive and negative because that's absolutely normative, I think. Thank you.
So, thank you very much for this excellent presentation.
Thank you.
And in continuation of our questions, do you think a protein has an umwelt? What is the smallest possible umwelt you can imagine?
Yeah, so when I first wrote that paper, as I said, I didn't talk about umwelt, and so when I couldn't sleep at night, it was like, does that arse have an umwelt? And I couldn't justify it, because by itself it doesn't. Even with the 20, which I was aware of at the time, it doesn't. But once it enters this functional, well, this loop, whatever we want to call it, that loop, Now I think the Umwelt resides in, well, I would say the Umwelt is in the loop, not necessarily the protein, but I'm open to change on that. Thank you.
Okay, thank you very much for this nice presentation. Yeah, there's like 1 minute, so you can do it.
Thanks. Excellent presentation, as everyone is saying. I subscribe. Really interesting. I don't have the knowledge on things like eyes to really pronounce myself, but it's a really interesting thing that Canguilhem doesn't let people sleep at night. Because, I want to reflect on this thing. Again, I just don't know the gene theory concepts properly to pronounce myself on this, but do we need to assume in this really, as you put it, Lorentzian way, that something like the Umwelt or the semiosphere must precede the possibility of semiosis? Wouldn't it help your argument if you consider that semiosis and its context co-emerge? Which, yeah, but not the Lorentzian perspective.
Sorry, if the semiosis co-emerges with
its context that enables it, let's say semiosis and Umwelt, semiosis and semiosphere, what we want to conceptualize it, that they co-emerge. Wouldn't that be a take as well, or your following your argument?
Absolutely, I think it co-evolves. It has to matter to someone. And so that's why I think semiosis co-evolves with normativity.
Right.
Thank you.